Welcome back, everybody. I know everybody's going to want to get on the road at some point, so it's probably a good time to kick off our afternoon session. And I'm going to speak a little bit more, briefly, mind you, but I guess, when I opened this morning, very much what I was trying to convey is, what we're about and what's in our current phase 2. And I thought it was timely as we open up the afternoon session to talk a little bit more, future and as we look forward. So I suppose I'm going to start with the sector. And, I think anybody living in this country is probably very well aware of just the significance and the size of the pharma and biopharmaceutical manufacturing sector in the country, and actually talked about it with a few people over lunch. I would say, when we look at what's happening globally, we're in the fortunate position through things like our corporate tax rate and so on that we're insulated, I think, from a lot of this. And I think that's largely down to significant sectors like the biopharma sector. So I have some numbers there where we've captured, I guess, both the multinationals, but also the indigenous sector. So we have both Enterprise Ireland and IDA represented there. And I guess thinking from the patient perspective then, Ireland is accounting for 40% of the medicines produced around the world. So, we were huge players, I think, as a country in terms of the COVID, and I suppose Pfizer as an organisation were pivotal in that. And huge employers, so 43,000 directly employed, and equivalent numbers really in the indirect, so thinking of the indigenous and support services. So that's huge in terms of not just corporation tax, but jobs and ongoing growth in the country. And I think, there is expectation around further growth. We see new investments coming in. I don't have all the details here. We do have a colleague from Biopharmaceutical Ireland talking with us later. I'd encourage you to look online for their latest strategy report that gives a lot of that data. So I just want to acknowledge that and to say, look, I think it's really important. We're really proud to work with this sector, and it's great for us and for the country that there's such great growth opportunities there. And of course, we're trying to be part of that long term and that longevity for the sector as well. So with that in mind, and thinking of our current research, we undertook, actually in the depths of COVID, to decide if we're an industry-led centre, we really need to figure out where it's going. So much of the world had changed. We started with a survey that we were supported really heavily with our partners, Enterprise Ireland and IDA, to go out to companies, not necessarily our membership, we wanted to go as wide as we could, and to begin to ask questions about current needs, but also future needs, so that we could try and, I suppose, take the temperature of the industry and to think about, right, well, what is it we do and how do we make sure that we remain relevant? So we did those surveys in Q3 2021. And then we assimilated the results. We had some workshops where we got many more stakeholders involved, and we tried to dig a little bit deeper to really get a sense of what it is that our mandate should be, and is it still as we understand it. So we had 19 multinational sites respond, so really good sample size. That doesn't represent every site, but it's a really good sample size. 36 of our SME members, so indigenous companies there, contributing, so really well represented as well. And we also went out to the HEI so we could try to understand within the university network today what's existing in terms of capabilities, both infrastructure and academic talent, just to match everything, to say, well, as a centre, if we're trying to serve both sectors and we're trying to leverage existing capabilities, are we set up? Is there any gap, anything that we really need to attend to? And of course, as a national centre, our focus was across Ireland. So we've generated a really short synopsis, a report that we're happy to share with you today. So hopefully you had the opportunity to pick that up as you registered. If you didn't, I'd encourage you to pick it up on your way out and read it or share it with your colleagues. We can distribute it as a PDF as well. And it's really how we've distilled down what we think is important and how we feel like we can really add value to the sector moving forward. And just, I suppose, summing it up very, very succinctly, because I don't want to take time in the afternoon session or from the afternoon session, we see that, small molecule remains strong. I think there's some that may have thought that this was a dying sector, but we're going to hear later on from a member company, AstraZeneca, who are, Rachel in particular is involved in the establishment of the new facility for small molecule, high potency small molecule in Dublin. So again, another example of growth. Digitalisation is probably, we know from our survey, it's on everybody's agenda. That's across Indigenous companies and multinationals, and great to see that that's a priority for everybody, because that's really rich ground for us then. And I guess we'll deliver competitive advantage for everybody, and we heard about it in the data analytics panel discussion. It's a bit like, if we're not data-led, folks are going to eat our lunch, and I think as a nation we have to and want to lead on this. There's a lot of initiatives already happening, so some small molecule leaders, again, working across facilitated through IDA, been working for about the last 2 years on this aspiration and trying to stand up initiatives around that. So within the ecosystem, there's a lot that we can do collectively. Data analytics, again, that's a foundation for the digital journey. We've already started some really significant work that you've heard about today, and we continue to grow that. This is the one area that everybody really, really wants to grow in, acknowledging that there's gaps there today, but also acknowledging that we have to get on the horse, so to speak. Continuous manufacturing is growing, and one of our academics, Emmett O'Reilly, will talk a little bit about that, and also in the large molecule space. And sustainability. So while this isn't in our shop today, it's inherently in a lot of the work that we do through cleaning, and we have a panel discussion on that before we close today, because I guess another area of really rich ground and opportunity. And I guess if we think of that, we have had the COP summit there recently, so this is on everybody's agenda. This is a global issue, and perhaps historically our sector has had a bad reputation, or at least a perceived bad reputation on that. Certainly has to be some low-hanging fruit there, I would say. So we'll look with a little bit of a more keen eye on that, and sort of whet the appetite with the discussion today on that topic. We would like to invite everybody here to a workshop that we'll do on that, so it'll get us into a deeper dive on that topic. It probably will be March timeframe next year. We'll send around some sign-in sheets, and so anybody who's interested, whether you're a member or not a member, we really want to garner as many insights as we can for our future focus. So I'll move on now, and I guess skills development, we talked about that the last time. It's really linked to all of the other topics, and this is where we see we can add value as we move forward. So I'm going to wrap up there, and I would like to welcome our first speaker into the afternoon session, and this is Dr. Emmett O'Reilly, and Emmett is a senior lecturer here at the University of Limerick. Let me just get your slides there. Right, over to you, Emmett. Thank you.
That's great. Thank you very much, Sarah, and thanks for the opportunity to come here and speak today. So my name is Emmett O'Reilly. I'm a senior lecturer here in the Department of Chemical Sciences in UL, also a PI here at the Bernal Institute. And today I'm going to speak a little bit about some of our work, which looks at droplet confinement for improved properties of both large and small molecule APIs. And all of this work we've done within a continuous manufacturing framework, which obviously is very relevant to the work that we do as an industry today. So given the audience that we have, I believe most people are familiar with the benefits of continuous manufacture. So we see improved batch consistency. We know it's a more cost-effective way of manufacturing our drugs. We know it's more sustainable. We also know that there's various processes which mean that it can be more scalable. So it's all around, it's a more efficient way of manufacturing drugs. What you're probably not too familiar with are some of the limitations of continuous manufacturing, and they often get overlooked in many kind of academic talks and also industry talks. But predominantly when we look at the control of polymorphism within APIs, bringing that within a continuous manufacturing process is a challenge, and that's some of the work that we work on in my group. So what I'm going to speak about today for the first part revolves around crystallization and confinement. And we have two approaches to this. So we use droplet confinement and also porous confinement. And the continuous manufacturing technique that we performed all of this work on is spray drying. So in terms of crystallization and confinement, why would you actually want to do this? Well, some of the technical benefits that you have, if you can control the nucleation, you have much more polymorphic control, so you can predict which polymorphic you're actually going to obtain. One of the main benefits is with respect to stabilization of metastable polymorphs. So we have a big issue with BCS class 2 drugs with respect to solubility. Many of them do have metastable polymorphs. However, stabilizing these metastable polymorphs is a challenge. So as a result, they're not typically used. We can also control the actual structure of the drug itself, so we can manufacture these in prism versus needle formats, etc. All this then translates into improved patient outcomes. So drugs with improved solubility, we're going to see enhanced therapeutic benefit and also reduced side effects. And if we take this approach and we look at multi-component pharmaceuticals, this represents the opportunity to address the patient pill burden. So the first part of this work, droplet confinement via continuous manufacture, this was published a couple of months ago in Crystal Eng Com. And it also won a few awards from the Royal Society of Chemistry. So really what I'm going to show today is just a synopsis of these results, but this is the article if you want to go and look at any of the results in a bit more detail. So before I begin, let's just have a quick recap of what spray drying actually is. So if you have taken a protein shake, okay, you've taken a spray dried product, same with baby formula. Essentially what we have is we have a liquid feedstock, okay, which flows into an atomizer, and as part of that we flow in hot nitrogen or an atomizing gas. This converts your liquid into an actual solid. This process happens incredibly quickly, and we actually collect the product then down in the actual below the cyclone. What we do in my group is we change the ratio of the atomizing gas, okay, to feedstock, and as a result we can alter the droplet shape. Okay, so we have two examples here. We have the larger droplet up the top and we have the smaller droplet down the bottom. So what this does is physically reduces the size that an API has to crystallize within. Okay, and as a result we see substantially different polymorphism. So this particular study we performed using carbamazepine, which is an anticonvulsant medication, and it's typically administered in form 3. So form 3 is the actual commercial form, however form 4, okay, is the metastable form, okay, and this does show enhanced dissolution properties in the metastable form 4 relative to form 3. Excuse me. So when we look at the table here, and I'm going to just talk through kind of some of the main results that we've seen. So if we run from sample S1 to S4, so 4 individual samples, we can see here that when we change the actual atomizing gas flow rate, we get a subsequent change, okay, in terms of the droplet size and also the particle size. And we can confirm this using particle size distribution, as you see over here on the right. With respect to, I suppose, the best graphical representation of what's happening here, that's down here in the bottom left. Okay, so as our droplet size changes, so does our final particle size, and we can see the corresponding SEM. We can look at the surface morphology. Now, this ordinarily isn't that exciting. You would expect some change in terms of final property parameters with respect to altering spray drying conditions. But where it gets interesting is when we actually look at the DSC results. Okay, so what we can see here is our crystallization peak for form 3, which is approximately about 190 degrees C. With samples S1, S2, and S3, we see a combination of form 3 and form 4. So remember, form 4 is our metastable form. This is what we want to ideally produce. However, we see a combination of both the form 3 and the form 4 for S1, S2, and S3 samples. And what we observe is that over a very short period of time, any of that form 4 that has formed transitions into form 3. Now, what's really unique is if we look at the smallest droplet size. And up here we can see that we actually have 100% form 4. Okay? And we initially were quite excited by these results. We put it on stability testing and took a look to see how the actual does the actual polymorphic form change over time. We've confirmed that with PXRD. Okay, and we can see that it remains stable after 3 months. Well, it's 3 months when I compiled this slide originally. It's actually 6 months now. So we've demonstrated a way to actually produce that metastable carbamazepine in the form 4, okay, which again has enhanced dissolution properties and potentially improved patient benefits as well. So what does this kind of mean? Well, there's a few benefits. So from the scientific technical perspective, it allows us to, for the first time, actually produce carbamazepine in form 4. And it introduces the approach of actually targeting specific metastable APIs. It gives us a certain degree of control over the nucleation direction in a continuous manufacturing framework. But I suppose with respect to the community that's here today, the real potential of this work is within, I suppose, producing metastable crystal forms for current APIs that exist. Okay, so we've demonstrated a method in which we can do it by actually using the size control, and we're rolling that out to different types of APIs at the moment. Another potential benefit, we're probably all aware of this patent IP cliff. For example, if we can manufacture the same API but in a metastable form as opposed to its conventional form, that's potentially new IP as well. Okay, so it's a way of prolonging the IP for existing APIs. So part B, leading on from this, we then chose to look at repeating the study, but rather than using a droplet size for the confinement, we actually went and used a porous structure. Okay, so we used porous silica in this case. And part of the reason behind doing this was, first of all, to develop a stage 1 screening method for future droplet confinement studies. So when we are processing this using spray drying, we do have a certain amount of control over the actual droplet size. Okay, however, we can get more precise control if we use a porous substrate. We also wanted to test the hypothesis that a pore size must be 20 times the molecular radius of an API, okay, for the API to actually crystallize within it. So to give you kind of an overview of where this work stands, and this is very much ongoing, we have 4 different pore sizes here, or 4 different particles with different pore sizes rather. We have a carbamazepine here on the left. So these vary from 60 angstroms to about 500 angstroms in terms of pore size, and the actual carbamazepine size is about 8.8 angstroms. Okay, so it should fit within, definitely should fit within each of these pore sizes here. We then made up a series of different loading ratios. Okay, so the loading ratio refers to the ratio of carbamazepine to your actual silica. And then we crystallized each of these products using 3 different techniques, okay, namely slow evaporation, rotary evaporation, and also our continuous technique, spray drying. Now, there's a lot of results here. I'm not going to show them all today. I'm really just going to focus on the kind of a synopsis of the results for the last 2 pore sizes that we looked at here, namely the 200 angstrom and the 500. So when we look at the DSC, and again we're using the DSC to confirm whether we formed the form 3 or the form 4, we can see that for slow evaporation and also for rotary evaporation, okay, we get a mixture of form 3 or 4, or we get pretty much all form 3. Spray drying is the only technique, okay, where we actually see form 4 being produced, and we see that the effect of pore size directly impacts the concentration of form, or the amount of form 4 you have versus form 3, as we see down here in the bottom right. So what does this mean? Well, in terms of pore size and technique, okay, we can see that both the pore size and technique have an effect, okay, on the actual phase behavior of carbamazepine. The porous confinement can be used as a method, okay, to obtain different types of polymorphs, okay, by tuning pore sizes. And ongoing stability tests will, I suppose, tell us over the next few months how stable or metastable polymorphs actually are. And using these techniques and these methods that we developed, we're now looking at a range of different APIs. So that kind of concludes just the small molecule part of this presentation. I'm going to move on now and speak a little bit about the large molecule side of things. So within large molecule, we see it's a rapidly expanding industry, and continuous manufacturing obviously is coming into that, and one of the main techniques for both drying and manufacturing biologics, okay, is spray drying. So number of benefits, okay, we see shorter production times, we see relatively larger shelf lives for the biological product relative to some freeze drying techniques, but more importantly, spray drying offers us the potential to engineer the particle. Okay, so why would you want to do this? Well, if we think about pulmonary delivery as a method of drug delivery, it's one of the more efficient methods compared to, say, oral or compared to transdermal or intravenous. So in order to manufacture a drug that has the specific properties for pulmonary inhalation, such as a dry powder inhalation, there's a certain set of criteria required. Okay, so for example, it needs to be below 4 microns in size. It needs to have a specific surface morphology. And spray drying is a technique that you can actually do that. Now, why isn't everybody actually spray drying biopharmaceuticals at the moment? Well, if we think about the process itself, it's a very energy intensive process. It involves high heat. The biologic that you dry, or any material that you dry, okay, is subject to a high degree of shear stress. And as a result, we often get protein denaturation. So you could spray dry a biologic, you could have the desired final properties, however, it's after losing any of its biological activity. So as a therapeutic, it's effectively useless. So what we do is we look for strategies for addressing that. So we initially set off in terms of our aims, and we've kind of expanded on this since. So we set out to develop a roadmap for spray drying stable dry powder biotherapeutics, okay, with retained biological activity. So retaining the biological activity is key here. And identification of critical processing parameters with respect to particle engineering for pulmonary delivery, okay. So ideally we want to be able to produce these particles with the right size for dry powder inhalation, but also have a very high level of activity retained. We initially chose lysozyme as a model biologic. So there's a lot of runs with respect to spray drying to optimize this process. So we needed something that's relatively robust, something that's relatively low cost, and we chose lysozyme in that case. So stage 1 involved spray drying lysozyme, okay, at low, medium, and high rates to actually evaluate the effect of both the temperature and the shear stress on the activity of the biologic. Stage 2 involved introducing an actual excipient. So in this case, we introduced trehalose at different ratios, okay, to see does this actually protect the biologic during the drying process itself. Stage 3 then looked at optimizing the process to obtain a biologic, okay, with a high retention of bioactivity, high yield, and high particle properties suitable for pulmonary delivery. So up here in the top right, we can see the SEMs from lysozyme, which has been spray dried itself. Okay, so you can see, depending on the parameters that you input into the spray dryer, you can control the particle size, you can make them porous, you can make them spherical, you can make them concave, depending on what you actually want to do. Where it gets interesting is in the stage 2 of the study, and that's represented here in this graph on the left-hand side. So within my group, usually we develop these process maps, and these essentially gives an indication of the sweet spot with respect to our processing or our operating conditions. So right here we have a plot of particle size, okay, as a function of trehalose or excipient percentage, and also atomization gas flow rate. And the bar chart here on the right-hand side, we can see particle size ranging from 18 microns down to 4, going from the bright red down to the dark blue. What does the process map actually tell us? Well, it says to us, if we want to produce a particle that has a sub-micron size, we need to be in this region here, okay? So what does that mean? Well, it means we need to have an atomization gas flow rate of about 740 litres per hour, and approximately trehalose or excipient content of about 40%. Okay. Now, this creates a little bit of a challenge because we also want to retain the biological activity. Okay. So where we actually stand is we have our biological activity retained here, and we then relate that to the particle size. So if I go back and I take a look at what we see, with respect to our enzymatic activity, this drops off very quickly. So if we process at 742, we're going to kill all the activity of the enzyme. However, if we plot the bioactivity, okay, versus trehalose content versus atomization flow rate, we can see that there's potentially a sweet spot over here in the right-hand side. Okay, so if we process within these particular conditions, okay, at 742 litres per hour, and also with the trehalose content of 40%, we can actually produce the biologic with the defined size for dry powder inhalation, but also something that actually retains the activity. So where does that leave us in terms of our conclusions? So the addition of the excipient trehalose results in a higher retention of bioactivity, especially at the highest atomization gas flow rate. And we've then gone on to further optimize this. So by processing at the conditions predicted by overlaying the process maps, we can achieve these particular parameters here, okay? So we can produce particles that have high level of activity, greater than 90%, size of about 4.1 microns, yield, relatively respectable yield of 54%, and residual moisture content of 3.72. So where we stand with this work is we are currently investigating a variety of different small molecule APIs, okay, based on the process I showed you in part A and B of the presentation. We're also looking at investigating these APIs specifically to target stable, metastable forms. On the large molecule API side, we're expanding away from lysozyme. We're now working with some large molecule proteins, and we're developing individual process maps for this. So where this feeds into the community that's here today, we're very keen to work with you on this. So for example, if you have a small molecule product that maybe it's a drug that's on the market at the moment, but you know there's a metastable form that potentially can benefit you from in terms of future IP or improved therapeutic outcomes, by all means come speak to us. We can run your samples on the processes that we've developed. Same thing for large molecule. If that's a space that you work in, we can actually go and apply our process maps, okay, to your excipient and large molecule protein concentration and identify what works best for that. I suppose from listening to me here, you'd be forgiven for thinking I've done all this myself. I haven't. I'm very fortunate to have 3 excellent PhD students, namely Alice, Laura, and Emily. And I should also thank Dr. Ahmed Zai. So a lot of this work is based on the work that was in his thesis, which he submitted back in 2020. Here's my LinkedIn profile. That's the QR scan, so you can scan that with your phones now if you want, and I'll add you to LinkedIn. And I'd also like to thank also our people that have supported this, namely PMTC, SFI, Bernal, Enterprise Ireland, SSPC, and also the Irish Research Council. So thank you very much for your time. It was great to be here.
Have you thought about the next steps for the polymorph, for what happens to it, or similarly next steps for the stabilized biologic in your spray drying system?
More so the small molecule side. So what we would like to investigate now is does that so it's on our to-do list. It's more the process maps for the large molecule, but it's a consideration, and that ultimately, if we are producing these metastable forms, we need them to remain in that metastable form as they go through all aspects of the downstream processing. So it's a future work, essentially.
Okay. Yeah. Well, shameless plug, we have a compaction simulator with unique capabilities at PMTC if you want to use that. So.
Sounds like we need to have a coffee.
Yeah, definitely.
Thank you.
Okay, great. And with that, I'll hand over to David Egan, Senior Research Fellow within PMTC. He's going to talk about some challenges in processing.
Okay, thank you very much. So I'll do my best not to wake you up after your post-lunch slumber. So this is a lightning talk, and I took the term lightning talk seriously, so I'm not going to be here too long. Okay, so what I want to talk to you about today is our new fluid bed capability and a coating case study that we're going to undertake. That we're going to undertake on this new equipment. So, within the pharmaceutical industry, there's a growing trend for more and more potent API materials, and because of their potency, these are dispensed at a low dose. So using conventional and traditional OSD manufacturing methods, it's not easy to produce good API content uniformity in a tablet. Okay, so what we are planning on doing is to use a fluidized bed system to coat an API or an API surrogate material onto cores, and we'll see in a minute what I'm talking about, and then use these in the remaining OSD manufacturing methods. So by distributing the active material or the surrogate material on these tiny cores, you get essentially little granules with a little amount of the API on the surface. So when you then combine all these together, you inherently get locked in your API distribution. Okay, so that will give you, and as long as your weight variability is low, you will get a good tablet-to-tablet variability, and also within tablet homogeneity as well. So with this project, we're planning on looking at two streams, a conventional manufactured stream and then a sprayed granule stream, and then producing tablets and looking at the homogeneity of API between the two. So here we have the two streams, which I'll try and use a laser. So on the top, we have a conventional OSD. And conventional OSD is where you dispense your API, your excipients and everything into a vat. You then blend or mix them homogeneously as best as you can, assuming they all flow well, and that's an entirely different kettle of fish. They go into a tableting machine. So it's not easy to produce a homogeneous mixture at a low concentration in powder form. And the two images on the right-hand side are just examples of, you can imagine that if you don't get a good homogeneous distribution of your API within, it could look like either of these two. So where you have areas of high concentration in one place and very little in another. So to try and counteract this, we are going to look at fluidizing compressible cores, something like cellets. They're essentially granules of microcrystalline cellulose, or the other option is to use sugar cores. And they're freely available on the market. So to make an API solution or suspension in liquid form is far more simple and more homogeneous than doing it in solid form. So the plan is to fluidize our cores, take our homogeneous mixture, solution or suspension, and spray it onto these cores. So you'll get an even film of our API material on each of the individual cores. So then when we compact them in the next compression stage, you get something like this here. So you have each of your cores are going to three-dimensionally, plastically deform and intertwine, but around the API is very much well distributed, inherently distributed well around each of the cores. So your tablet at the end should be far more homogeneously distributed within each tablet. And as long as you have a good weight distribution, you should have a good weight, a good homogeneous API distribution between tablets also. So how will we plan on doing this? Well, we were very lucky to have won some funding from an EI Capital to be able to purchase this GLAT multi-lab system, and that is capable of granulating and Wurster coating materials and also drying those materials within itself. So this is an advanced manufacturing platform, so essentially all the process and sensors and PAT technologies are brought in to SmartX, which is the Inopharma system, which is built on top of this GLAT unit to actually take care of all of the input streams to be able to build models to control the system. So we'll see a bit more here. So as I said, all the process and PAT data streams are accessed using the multivariate interpretation, and these are used to build models. So, for example, you can have your airstream temperature, your airstream rate, your product temperature, etc., all as process. You could set those, and you could stream all that data in real time. Now, in addition to that, we also have PAT technologies on the system, which give us particle size analysis. So we could have particle size analysis in terms of our D50 and our D10 to D90 to be able to monitor that while we're actually spraying, and we're also able to build models from our NIR input data, and these can be deployed in SmartX so that we can have a real-time analysis and determine other properties as we see next. So, for example, this graphic is work done by Sam and Marcus and Chris in Inopharma, and it shows the evolution of the particle size, which is the yellow line on the bottom, within the powder bed. They use the fluidizing airflow rate to maintain fluidization. And so that was the feedback loop to actually produce these materials. But on top of that, they were able to use the NIR data to to model the actual moisture content and predict that, which is the orange line. And at line, they were able to take samples and undertake loss and drying analysis. And you can see that they actually match the NIR quite well. Okay, so this shows that the system is able to feedback control with inputting process data and also from PAT technologies. So the unit operation is capable. It can do both top spray granulation, where you would take a fine powder. Again, as I said, in terms of flow materials, if something has a bad flow or you just want to granulate it, you can add in the finer materials at top spray, and you'll produce larger granules, which will flow better. But also, and what we're going to use is the bottom spray Wurster tube, where we can put in our granules and actually bottom spray and coat them in an even way and produce. And our plan is to use all the PAT technologies to get an end of spraying set point and automatically actually stop when we have the correct amount of API material or surrogate material on our coated material. So, as you can see, we now have a unique resource to help develop or improve existing fluid bed processes. And this work that we're going to undertake, we should show the unit's capability and generate a case study which will demonstrate the feasibility and the power of the real-time data and data analytics in supporting development. So, as I said, we can fully monitor, we can model, we can build control strategies for granulating and coating operations. And if there's of any interest to anybody, feel free to contact us at any time, and we'd be delighted to work with you to benefit your processes. Okay? Thank you.
That's great. Thanks a million for that, Dave. Any questions for Dave before we move on? Okay, so I'm delighted now to welcome a member company, AstraZeneca. So we have two colleagues from AstraZeneca who've travelled from the UK, so really appreciate that. And we're really excited to hear about the sustainability case study. So welcome to Andy Barnes and Rachel Dawson. Let me just open up your presentation for you. Over to you.
Thank you, Sarah. Really pleased to be here. I'm very grateful for the opportunity to talk you through what started life as a simple project as part of our continuous improvement program for our cleaning process on the pilot plant, and through a disaster during that project transformed itself into quite an impressive sustainability project. I'll take you through I'll give you an overview of the pilot plant facilities at Macclesfield and show you our cleaning process and how we came about to identify this particular process. And then Rachel will take through how we embedded the process onto the pilot plant and talk you through the outcomes. So drug substance manufacture is based at Macclesfield, AstraZeneca's Macclesfield site. It's part of the clinical manufacturing supply chain, and we manufacture development compounds for the clinical manufacturing program. We've got a multi-product facility manufacturing a range of API intermediates for the clinical trials. We have 6 production trains across a number of plants, ranging from 250 litres to 6,300 litres. We have a hydrogenator and a dedicated Pures facility. And of course, it's an old aging plant. So some of our plants are greater than 35 years old. And that means that it is a really intensive process to clean the plant. Cleaning from small molecule API is difficult, especially in the development field, because we don't necessarily have an understanding of the mass balance and the limited information available to us to target specific cleaning activities. So we use solvents predominantly to clean our plant. We do use detergents, but as a last resort once the solvent process has failed to achieve visual cleanliness. So we use solvents to dissolve the remaining materials, and then we do not have a treatment facility on site. So all our effluent, whether that's a drummed waste stream or bulk effluent, is taken off site for incineration. So although we're a relatively small facility, this is responsible for up to 30 tonnes of waste per year, and that corresponds to approximately 50 tonnes of carbon dioxide per year. So, small molecule API plants, they're really complex, and we have to clean them in a modular process. At Macclesfield, we rely on condenser flushing capability, where we can pump the cleaning solvent to the top of the condenser head and flood the condensers and the reflux return vapor uplift, and then refluxing the cleaning solvent in the vessels. All our filter dryers, pressure filters, are fitted with spray heads, but they're not impingement heads, they're just wetting heads. And just one of the equipment train is fitted with spray heads within the reactors themselves. The rest of the equipment trains, we rely on condenser flushing and refluxing of the clean, so it is really quite resource intensive to get through to the acceptable carryover. So we apply a modular process. The first thing we do is get rid of the gross contamination, and that's always the slurry transfer line from your crystallizer through to your pressure filter, the pressure filter heel, and then the filtrates line coming back out to the receiver. So the pressure filter heel, for those that aren't familiar with the pressure filter dryers, they have a Hastelloy stainless steel cloth that's usually 20 micron, and that's the solid is captured on that, and we have an agitator in the pressure filter to make sure that we have a homogeneous mixture. We have to be extremely careful that we don't rip that cloth. If we rip the cloth, the batch goes through to the receiver. So we always set the travel of the agitator to 1 centimetre above the cloth, and that 1 centimetre of product between the base of the agitator and the cloth is called the pressure filter heel. Some of the filters we can recover that heel, but usually that's remaining, and we have to dissolve that. We then remove the what we call glove bags. It's our flexible containment, so that's how we charge the solids into the equipment and the discharge liner from the pressure filter chute. And then we go into a series of solvent refluxes and condenser flushing. So we do at least 2 of these cycles in each equipment train, and this can range up to 1,000 litres each time. We then have to prepare the unit for our module 2. So we, at that point, we break into the hard to clean areas. So we need to ensure that we haven't got a flammable atmosphere, so we stick a water wash through the whole equipment train to remove the solvent. We then target the hard to clean areas. Our larger plants, they can have in excess of 80 breaking points where we physically dismantle the plant, inspect it, clean it as required, inspect it to formally sign off that part of the equipment train is visibly clean, and then we reassemble, ready to do our final rinse wash verification analysis. And because we've dismantled the plant, we have to do a water wash again, just to demonstrate integrity. It's a lot easier to contain a water leak than it is a solvent leak. And then we go into our final module, module 3, and that's the rinse wash verification process. And that will take at least 2 full refluxing solvent washes throughout the whole equipment train. So again, there's another 2,000 litres of solvent as a minimum going through. Once we've demonstrated that we've met the acceptable carryover, we vacuum dry the equipment train and visibly inspect for cleanliness. And this is where our cleaning or continuous improvement strategy came in, because on average we use 3,500 tonnes of solvent, 4,000 tonnes, 3,500 kilos, 4,000 kilos of water, and the average cycle time for that whole process is 3 weeks. We've got examples where that's taken an awful lot longer. So we wanted to understand how we could reduce those. I was aware of technology that was developed in the Astra API sites over in Sweden, and it's called the pica cleaning process, and they used activated carbon and recirculated the rinse solvent rinse wash through the carbon, reducing and removing the solute, and that led to a significant reduction in the volume of solvent required to clean the plant, so much so that they achieved a greater than 80% reduction in the solvent required to clean the plant. Astra liked it that much that they named it after the two engineers that developed it, and that's where the pica comes from, Peter Lindquist and Patrick Carlson. We have historically used the pica cleaning process at Macclesfield. The technology was transferred from Astra through to our parent, our sister sites, when we merged and formed AstraZeneca. And this is a cleaning rig that we inherited from one of the sister sites and used at Macclesfield. It's very difficult to just scale on that photograph, but the handle on that cage sat this high with me. So using it at Macclesfield, it was a lengthy process, and we just historically didn't see the same level in reduction of the cleaning solvent as the Astra site had had. And the skid occupied a large footprint and required a separate GAF filter to house the activated carbon. In our case, we used pelletized carbon norit. And then, with it being, we're embedding the process into aging plant, we needed a large number of flexible hoses to make the connections, and then make and break the connections to change the specific target vessel as we stepped along the process. And perhaps the main reason why we didn't see the same level in reduction as the Swedish plants did was because when we first trialled it, the cleaning process was only used to clean the reaction vessels. It wasn't used to clean the pressure filter, which we know is the area of the largest hold-up within the equipment train. So we embarked on a number of trials. I'll get to the photo in a minute. So we have, with being a development facility, although we're continually manufacturing API, that is not necessarily one campaign following another in the same plant, and we quite often have a little bit of downtime in a plant, and we can use that time to trial cleaning initiatives. We targeted our smallest plant, the plant vessels fitted with the spray heads, and also with the condenser flushing capability. And this is also one of our newest plants, so we have routing stations so we don't have to make and break so many connections every time. We used water, and we determined the minimum recirculation volume for each vessel and each combination of condenser flushing and then recirculating via the vessel spray heads. And we started in the smallest vessel first and then stepped the cycle up right the way through to the largest receiver. And then we targeted, can we use the cleaning skid with the pressure filter? And we demonstrated that we can successfully pump from our receiver back through the slurry line into the filter and then return it back in a single continuous system. And that was the proof of concept that we needed. So we then started to consider how we're going to embed that into an actual cleaning process. And this is when our engineering team sent me this photograph. So that's the frame of our cleaning skid, and as I mentioned before, it was at least 25 years old when we brought this back online, and engineering, certainly welding standards, are not what they are now. And unfortunately, the welds had given way, and we were unable to take that forward. And there's our disaster. We prove concept, and then we lose the equipment required to run it. So Rachel and I, we went into a period of reflection, and we understood that we'd achieved something with demonstrating that we could get through to the filter. But we also, once we analysed and took a step back, we realised that although we haven't got the skid, the pilot plant had all the components of the skid sat there just in its own right. We have pumps, we have an array of filters, in our case we have the inline UV. So we started to build a picture of how we could use the existing plant and apply the philosophy of peaker cleaning using the existing equipment. And then when we really started to think about it, we thought that we could use a different type of carbon. Instead of handling active loose carbon, we could go to a supported medium. And in our case, we used a Kuno filter, and 3M provided us with supported carbon filters for use with the Kuno filters. That significantly reduced the operational hazards associated with handling loose carbon. And then we focused on the actual solvent and the volume of solvent and how we would use the carbon within the cleaning process. And if you remember our modular cleaning process, the heel dissolution from the pressure filter, so our pressure filters range from 0.4 metre squared base area up to 3 metre squared base area. And that 3 metre squared base, you can have a heel of greater than 50 kilos. So that's going to require an awful lot of solvent in its own right to dissolve. But if you apply that to the carbon filtration to that, the number of filters or the amount of carbon required to remove that level of solute would be very high. So we're always going to have to dissolve the heel anyway. One thing that we do is we look to dissolve the heel, and can we use that in downstream processing rather than just send that off to waste. So we dissolve the heel off the filter, we remove the flexible containment, and now we start the solvent cleaning. Where instead of refluxing the solvent, we now have the carbon to help us, and we start in the smallest vessel and step up, working our way through the equipment train, and we recirculate the cleaning solvent through the carbon bed, and we use the UV to tell the inline UV to tell us when we've hit our target. And in this case, we applied the acceptable carryover quantity for the whole equipment train to the vessels individually. So we started in the smallest vessel, and we recirculated via the vessel spray heads, and then condenser flushed, and then we always completed the our approved cleaning step, our solvent reflux, at the end of that process. So we demonstrated that we hadn't impacted our approved cleaning process. So we have a cleaning solvent in our smallest vessel that meets the acceptable carryover quantity, and then we stacked that onto the next largest vessel and repeated that through to the receiver. Once we'd cleaned the equipment train, then we targeted the pressure filter, and we targeted the slurry transfer line in, the filtrates line back out again. And once we cleaned those to below the acceptable carryover quantity, we successfully transferred or recirculated the cleaning mixture via the pressure filter spray heads. We demonstrated with that process, we'd have the carbon remove the solute, we'd have rinse wash analysis of that one cycle that met the carryover quantity, and so we completed that one cycle with less solvent than it would have been for one cycle rather than the two refluxing cycles. We complete the water wash. We always go in and target the hard to clean areas. Water wash again, and then this is where the possibility of the real saving comes in. So you've got a 50% reduction in solvent for your module 1, but because that solvent has been cleaned by the carbon and it meets the acceptable carryover quantity, we reuse that solvent from module 1 in module 3, and again start with the smallest equipment train, smallest vessel in the equipment train, step ourselves through in the same process. The carbon is always working, removing the solute, and at the end we always have, the target would be that the rinse wash meets the acceptable carryover quantity, and in doing that you've saved yourself another 2 full volume solvent refluxes. So we're targeting a 75% reduction in the volume of solvent required to clean the process. So that's the theory. So now I'll hand over to Rachel to show you how that happened, or what happened next.
Yeah, great, thanks Andy. So yeah, as Andy explained, we had an unfortunate demise of our Pico cleaning skid, but we still had the targets to get to that 50% solvent reduction within our area as a business. So we kind of pulled together existing equipment we already had on plant. So this photo here shows you the setup of the new redesigned skid. And from right to left, I'll just go through. So we have the UV vis, which you can see on the right-hand side, an ATEX UV vis instrument, which is spool mounted onto the outlet of the Cuno filter. And the Cuno filter is there in the middle, and that hosts the carbon cartridges, which we use to absorb the API. With the UV downstream of this, we could monitor the remaining waste API in the solvent after it had gone through the filter. And then you can just about see it, but to the left where the flexible hose is, there's a downstream PAL filter, and this was put in so that we could entrain any loose carbon that would come off these filters to stop that getting into the plant and just being something else we need to clean. So this setup was verified on 2 cleans. So we started off using this on our smallest plant on product API 1, and we used an acetone IPA water mixture. And then we trialled this again on a second product, API 2, on our largest plant with a methanol water mixture. So that's how we kind of repeated the solvent savings on another plant. I was going to say next slide, please, but I'm in control here. So the first thing we need to consider when we are using the carbon cleaning skid is how we're going to recirculate this around the facility and how we're going to hit those process lines that we used to manufacture. So as Andy went through the cleaning process, so we'll target the vessels. So this is a diagram showing our smallest facility and how we set this up in practice. So first to charge solvent to the vessel to clean the vessels, we'd have to charge a solvent volume that would give us some buffer within the vessel, but also take into account the volume of the filters, volume of the GAF filter and all the pipe work associated, because we still needed to be able to take a sample of this solvent whilst we were cleaning. So we'd charge the vessel, we'd then pump it and recirculate it through the 2-pot GAF, which was already in place on the plant, so we utilised that, pass that through the Cuno filter, and then follow the flow around, and we'd hit the spray ball first to do the vessel coverage, and then we'd change the routing and we'd hit the condenser to target condenser cleaning. This diagram in particular shows a representation of us cleaning the pressure filter, which on our smallest plant we were able to do, but it was tricky in practice on our largest plant, where the pump wasn't capable of maintaining that pressure on the recirc loop. So what we had to do there was fill the pressure filter with solvent from the vessel. And when we had that volume in the pressure filter, then nitrogen blow transfer back, and then we could recirculate that through the skid. So that was some learnings for us there. So there's quite a lot of pre-work that goes into running the carbon skid, mainly around selecting the appropriate carbon grade. So we worked really closely with 3M to look at the different carbons, because they all have different properties based on their activation types. And they were able to give us some carbon discs, which we could put in the lab, and we had a setup of a syringe pump in a carbon disc filter, and we had a UV probe in line there, so we could assess how well the API was absorbed from the syringe pump, which was a waste stream that we took out. It had absorbed through the carbon disc, and this graph here shows R55, R54 carbon grades, and the UV probe was to look at when we achieved breakthrough. So when we'd have to change that carbon over. Secondly, once we picked a grade of carbon, there was then flow tests. So by optimizing the flow and the residence time through the filter, it also affected the adsorption capability. So that was another thing we had to look at. And this slide, this gives you an idea of what the operators can see on plant. So it is the operators running this UV as part of the batch sheet instructions. And so talking about the skills gap earlier on in terms of training them how to use this PAT was probably my biggest challenge on this. But on the left-hand side, you can see this was our first clean with the API 1 on our smallest plant. And we have an acetone peak, which does unfortunately kind of blind the response of the API, but you can just see it sits on the shoulder of the peak. That API reducing as we were recirculating through the bed. And to the right-hand side is our API 2 on our largest plants. This was the methanol water mixture. So there's no blinding of the data, so you can see a really clear reduction in the API in the waste stream down to 2.5 mg per litre. Unfortunately, that was the lowest detectable level of our UV instrument. We didn't use it for inline decision making, but the benefit to the operators was that we could see at what point we were becoming close to clean. So then it reduced the sample burden massively, so it helped in that instance. I'll just quickly talk to this. So some offline analysis that our chemist did in the lab was looking at the time versus concentration. So this big peak on the left shows you the reduction in the concentration over time, but when we correlated that to a batch sheet, we could see that that's when we were recirculating through our spray ball, and it took 35 minutes approximately to get down to those undetectable levels of our UV instrument. And then again, when we did the condenser flushing, you can see two little peaks there. So this is the impact slide. So looking at the percentages saved with our smallest facility and that product API 1, that percentage, 42% solvent, correlated to 1,000 kilos of solvent saved in that clean. And then on our largest facilities where we saw the greatest impact at 70%, so we saved 9,600 kilos of solvent in that clean using this technology. And then we also did a cost analysis on this, because obviously you've got the carbon filter costs, they're not cheap, versus the solvent costs. And the larger the plant, the more gross contamination you've got in that plant, the more cartridges you're likely going to use. So in terms of a cost analysis, there wasn't a cost saving that was a driver to do this. It was more around the sustainability metrics. So we had a sustainability assessment performed, and it showed us that on our largest clean, we saved 14.6 tons of carbon using this system. And it sounds silly, you're using carbon to save carbon. So that's what triggered the sustainability assessment. And if you extrapolate that across DSM for the year, it's a potential saving of 32 tons a year. A third party did this life cycle assessment for us, and the light blue colouring on these pillars shows us the savings with this new cleaning method versus our old cleaning methods. So it helps us see our environmental impact. So the learning. So I was very much a part of this project from the start through to the demise of the Pika skid, and then obviously there was various iterations because we were learning as we were trying to set this up. And these learnings we've now applied to the design of a new skid, which is being funded by an internal group at AstraZeneca called the National Resources Reduction Group, and they're there with their dedicated funding to fund these kind of sustainability projects. So we looked at whether a single pass through the filter versus recirculation around the filter would help adsorption. So the new skid allows you to do both. We can flexibly relocate the UV depending on the process and depending on what we want to look for. We've added a temperature probe to monitor the temperature effects on the UV. And during the commissioning, we had quite a lot of issues with pulsating pumps. So we had air-driven diaphragm pumps. We've now look to use centrifugal pumps and also to allow that variable speed flow. So that enables us to hit the flow rates on the spray heads as well as reduce that down when putting it through the carbon to hit those residence time for adsorption. And there's some level of automation included as well. So this is the new skid design to be delivered end of 2022. So when this is delivered, there's a bit of rollout to do in terms of getting this as BAU on the pilot plants. So they'll have, as Andy mentioned earlier, any one of our facilities has spray heads, so we'll have to install spray heads across all facilities to get the maximum benefit out of this. And also where we have simultaneous cleans, there's also an argument that we probably need a second skid in the near future. So I used to work alongside Andy for 4 years in the pilot plants. I've recently moved on to the design of the new API facility that's going into Dublin, so doing the process design on that. So we're looking at taking the learnings from all this work done on the pilot plants and applying it to our new facility to basically aim to reduce solvent usage at point of use. So we've allowed space for 2 of these units because we'll have 2 batch trains within our facility. And we've also been able to design the plant with cleaning in mind, which I think came up earlier as well. So we've got a gravity flow through the facility over 4 floors. We follow the process vertically. And we've looked at layouts of the equipment to reduce the cleaning. So where we've got crystallizers, they'll be directly above the filter dryers, for instance, so that's a direct drop. There's fewer hard to clean areas. We have multiple spray heads per vessels, hopefully, and we've got a lot of recirculating SIP lines to really hit every point of use in that process line. So yeah, we've also got, as Andy mentioned earlier, the flexible routing stations, so that allows us to either use this carbon technology or as future technology moves in, so membranes, for instance, we'll be able to swap this out as our knowledge of membranes and other types of technology develop in the future. So that was it for me. Thank you. Any questions?
Thanks, guys. That was a really, really insightful talk, and I'd like you to maybe take a seat and join the panel discussion. Now, you're not off the hook yet. So, you know, as I mentioned at the start of this session, we're looking towards sustainability, and we knew that the Macclesfield site were doing this work, which, of course, interestingly, and I don't think, Andy, you brought it up in this presentation, but I know when we spoke before, you had mentioned, you know, looking at that project on the traditional return on investment, it didn't have legs because it was taking more time. But when you look at it through a sustainability lens and carbon footprint, it's a game changer. And I think it's in that context that we really wanted this really insightful case study to be shared. So thank you so much. And it's a great segue into this panel discussion. So I'm delighted to welcome Padraig Summers, who's currently our chairperson, but actually one of the founding fathers, I would say, of PMTC. And so, you know, Padraig's had a long career in the pharma industry, both at drug substance and drug product, and been a site director, and so understands very, very well what the challenges are, and I suppose ever increasing challenges. Also coming back is Dr. John Costello. Or Cosgrove, sorry. So, and I'm delighted as well to welcome Michael Gillen. So Michael has, I think, planes, trains and automobiles were involved today to get Michael here from a meeting in Europe. And Michael is a senior executive with Biopharmachem Ireland with particular expertise in the area of environment, health and safety, and is also, I guess, a conduit for the sector between here and Europe to help us understand what are the new regulations coming down the tracks. So I guess with Rachel's design expertise and Andy's process expertise and the others in the panel, I'll stop talking there and I'll hand over to you. Oh, actually, too much talking. What I'd like to do maybe before we start, just so that back to the audience, both virtually and here in the room, using your QR code, we might just ask you about your own company, or does your company have an ESG policy to meet target of net zero commitment by 2030? We're just trying to get a sense of the degree to which this is really strategically important in your organisations. So we just get a flavour of that. I think it helps the panel. So that's a really, I suppose, good. It's 75%, even more again.
Looking very good there. Good. Work.
Yeah.
Okay.
Okay.
It's a moving target.
Happy.
Do you want to just talk about the capex, or do you want to get into it? So we had one other question. That's changing all the time. There, actually, it's nearly getting to 50-50 now. Maybe we'll move on to the next question just very quickly. Does your company apply sustainability factors such as carbon footprint into decision making like capex? And I think that's really just to try and get a sense of, we've seen that within the AstraZeneca organisation, that step change has happened, and hence this whole rollout and investment to really embed this kind of methodology. So that's really interesting, actually. That's looking like that's not mainstream as yet. And so maybe even some of those nuggets around influencing that idea within the organisations would be good. And I will stop talking there and hand over to you, Padraig, to chair.
Thanks, Sarah. Yeah, we'll let that settle down there for a minute. Yeah, this is... I've been looking forward to this discussion because certainly from where I'm sitting and over the many years that I worked in the industry, we have seen significant change in terms of the, I suppose, the discussion around sustainability. I suppose when I started working in chemical APIs back in the 1980s, the hot topics at that time were centred around odours in the harbour area, Cork, and caused a lot of grief for the whole sector at the time. And in some ways, it led to the foundation of the EPA back then. But things have moved on, and I think it's become much more of a positive discussion since then. And most companies are looking at the sustainability of their operations, which essentially is looking at the longevity of the business, or the potential long-term future of their company in terms of its ecological impact, its societal impact in terms of the people who are working there, and its impact on the environment and the customers that buy its products. And of course, then its economic return as well. And these are all very important factors. But what we're seeing and what's coming is that these things are no longer optional. They're no longer, nice to haves. We know that the investment community are looking carefully at companies' ESG policies now and saying, if you've got clear policies and demonstrate performance, then we'll buy your stock. And these investment companies are the guys who are investing our pensions, and they don't play with that money easily. So they're looking at better returns, and they're seeing better returns from the companies that actually invest across all the three headings. So we're moving from optional to obligatory. Our government has signed up to commitments under COP, and these are legally binding, and they will translate onto obligations for companies in Ireland and elsewhere across Europe. And I suppose, that kind of tees up the first, I suppose, question and topic, which I'll direct it towards Michael, which is, and again, Michael has seen the evolution over many years of these areas and how they've been regulated, and also seeing what's coming, because he's actively involved in various European groups looking at these. So I suppose the question is, and I suppose I should say before passing on to Michael, part of this is a lot of the discussion is very, we'll say, fuzzy. It's very high level, and it's not clear that companies, and certainly, other than maybe the large companies who are quite clear in this area, it's not clear that a lot of small to mid-sized companies are very clear about what they should be trying to do here, what they should be measuring, what they should be trying to achieve. And it's into that space that perhaps this is something that PMTC should be looking at in terms of supporting its members. So the purpose of this discussion is really to maybe tease out some of the topics, some of the unknowns, and maybe from that to frame an agenda for a more detailed deep dive, as Sarah talked about, next year. So maybe, Michael, starting with you, looking ahead, I suppose looking both at the companies that you were liaising with in Biopharmachem, here in Ireland, and also what you see at the European level when you're attending meetings such as the one this week in Bilbao. What's changing and what's going to change, what's coming, what should companies be aware of that's going to come down the road in the next couple of years, or maybe what's already there and they don't even know about it?
The first thing I'll say is I'm working in fact, Padraig and I worked with one another 30 odd years ago in a small little SME up in Baldoyle Industrial Estate. And my introduction to EHS was on the 9th of September 1993 at about 20 past 10 in the morning, when we had a major incident and we shut down the industrial estate. And that coincided with the year the EPA was established. And they were all over us like a rash. And that was my first introduction into what subsequently became known as sustainability. And the thing that has struck me over the intervening 29 years is, and I was really impressed with Sarah's figures when she started going, 90 billion. In fact, last year we exported over 100 billion of products from Ireland. Like, it is savage money. The reason, we were able to look after our citizens so well during the pandemic was because of this industry. Okay, so be in no doubt about it, this is the cash cow in Ireland. And the thing that scares me most about it is most of the people working in this industry do not realise that we're less than 0.1% of the chemicals being used in Ireland. So we think, we're a really important chemical industry. People know what we're about. Lads, you're going around with your heads in the sand. Because there was a chemical sustainability strategy published last year. And when you actually go through some of the things it talks about banning the most harmful chemicals in consumer products, allowing their use only where essential. Who's defining essential? Is it you? Is it the HPRA? And I got the tail end of the conversation this morning in terms of the data analytics and the importance of engaging with regulators. Unfortunately, it's not. It's consultants working on behalf of the Commission who determine what essential is. So until such time as we wake up and realise this is going to be the future of all of the jobs you're talking about sustaining over the next 10 years, you need to make sure that the powers that be know how critically important this industry is from a chemicals use point of view, because we use so little of it. Those of you who've been engaged with REACH, and there's a REACH reform process taking place at the minute, and one of the big things there is they're going to try and phase out more harmful chemicals. And one of the things they're targeting are a class of chemicals called PFAS, which are poly or perfluoroalkylated fluorinated alkyl substances, right? Now, when we think of PFAS in firefighting foam, bad, persistent organic pollutant, sits in the ground forever, not good for society, not good for our environment. But when you look at the definition of PFAS, it includes anything with a CF2 or a CF3 in it. And that's over 20% of all of the medicines on the market globally. So because we're allowing others determine and set the agenda in terms of essentiality of PFAS, we run the risk of actually doing ourselves out of a business. And it's interesting, in his introduction when Padraig said, the investors are now getting all over this sustainability agenda. This ESG is so important in making investor decisions. We have been regulated since 1993. And in fact, the local county councils regulated most of the plants that had emission licenses prior to that. So that's been going on 29 years. We have been regulated. What has happened in the last probably 5 years is the C-suite have now been asked hard questions by investors and pension funds. What are you doing on it? And within companies, there's a disconnect between the C-suite knowledge of it and the environmental health and safety manager on the ground level. And they're not actually communicating within organisations. Because when you're putting up the polls there, and one of the thoughts that struck me when Padraig was talking about the question you were going to ask is, how many of you have an ESG policy? How many of them work is the more pertinent question. Because it's very easy to put something down on paper to make you look good for your next investor roadshow. But here's a question, right? So for those who emit greenhouse gases, right? There's a scope 1, which is the greenhouse gases you emit. It's covered by your environmental protection license. There's scope 2 then, which is when you buy electricity, and it's whatever CO2 emissions are related with electricity. Over 95% of your greenhouse gas emissions, though, come from scope 3. The emissions in your supply chain. Who has even started looking at that? And are you making progress on it? Because that's where you're really going to be judged. There's such a drive within Europe to do away with harmful chemicals for the right reasons. But we run the risk of not articulating our position well enough and being caught in the crossfire. Those of you who, and I know within Ireland we have a lot of companies who had to file for authorisations for things like DMAC and DMF. These are two polar aprotic solvents that you only use when all the other ones have stopped working. If you're using them, and because it's a boiling point of 150 plus and 160 plus, trust me, you've tried everything else. So when you're using those, it's the land of last resort. There was an authorisation process put in. It costs an awful lot of money to go through that process. Currently, under the REACH proposals, they're talking about merging that with restrictions. You might find you'll not be able to use common solvents, chloromethane, dichloromethane. Could be a thing of the past within the next 5 years. How many of us use it? It was ubiquitous in our day in Newport. So my ask is, start working on the comms. Sustainability is new. In fact, sustainability, I think, is the love child between environmentalism and OPEX. Because I was listening to your presentation and I thought it was so good to hear you're doing more with less. That was the basis of OPEX for so long. But now we put the label of sustainability on it because it allows the C-suite sell it a little bit better. So start revisiting the comms internally, because there's a serious valley of death there at this moment in time, and it will cost us in the next 5 to 10 years.
Thank you. Just being specific then, our company is going to have to, at a site level, report on all the 3 levels of scope emissions? That you're aware
No,
of?
but what they're being asked now by the sustainability champion in the companies, the EPA are so far behind the curve on this. Because they will wait until a directive or a regulation comes out from Europe. But at that stage, in fairness, corporate is moving quite rapidly ahead on this. For the very reason you outlined, they're being driven by the investing community. So Biopharmachem is a part of IBEC. In IBEC, when they talk about sustainability, they actually talk about sustainable finance, which is actually the root of all sustainable conversations. Are we making money on it? If we're not doing it.
John, do you have any thoughts that you want
That's
to throw in the top of
this
that?
one. I may as well go home now.
Before we move on.
I'm glad someone got to the rant before I did.
Well, I know you want to. So.
No, I fully would concur. I'm not in the environmental space, but in the energy space, the Scope 3 emissions are the big deal in the supply chain. And if you're a supplier to a corporate who are going to develop net zero policies, as some have for 2025 or 2030, then you certainly need to be looking at, your Scope 3 emissions and all of that. I suppose where we've worked with companies is more on the energy side, and this industry has been fairly well organized in terms of energy management and energy management policies and managers or directors. But there's still an awful lot of low-hanging fruit. There's still an awful lot of value and constant. And for me, and as to tie in with the discussion earlier on, this starts at a digital level, because if you can't measure it, and if you can't measure it at the building level, at the production line level, at the batch level, then you can't really manage it effectively. And so for me, it's very much a process of being digital first, then applying lean, reducing waste, reducing energy consumption that isn't necessary, and then it becomes greener.
And do you think, is there enough clarity out there as to, from across companies, that they're clear as what are the things we should all be measuring and maybe sharing and benchmarking each other against? Is that an open area, for example, that PMTC might be able to support in?
Yeah, I think there's quite a lot of we'll politely call it confusion. There's a lot of greenwashing. There's a lot of initiatives, driving various targets, which, like some of our own national targets, we're going to miss them by so far that they're meaningless anyway. So, and arguing about whether it's 28% or 30% is sort of pointless when you're not even going to make 12. So it's you do see that in companies. There's certain flags on outside the building or stickers on the website, You know, all of which is good. I mean, it's all about communication as well, but people have to achieve real stuff on the ground. And I was reminded by someone recently, like, at the moment, the climate activists are targeting, you know, fuel companies, fossil fuel companies, but pharma is responsible for a huge quantity of water consumption. You know, we find that a strange one in Ireland because it's falling out of the sky all around us most of the time. But across the world, water conservation is also a huge topic. And, you know, you may find soup being poured over your brand by climate activists soon because of your water consumption. So that's also, you know, I think critical to the discussion.
On the water, and I think this is an interesting thing. I remember, so again, sustainability isn't new. So here's a quiz question for the people in the room and probably online. Next Friday, 2 weeks, is the 2nd of December. Can anyone tell me what happened 38 years ago on the 2nd of December? So it just shows you how short the corporate memory is, because that was the night of the Bhopal disaster. And the following year, when the Canadian chemical industry launched the Responsible Care Programme, that's when we started as an industry. So in 1985, we started addressing things like water use and waste use and so on and so forth. And I remember we used to run awards here in Ireland. And I think it was in 2010 or 11, and I see McNapier up in the audience there, when they were building what was Centacor at the time, they started to harvest the rainwater to run their grey water on site. Now again, how many of the plants now are even contemplating doing that? And it's a simple thing because it's a huge volume, like we're flushing it down the jacks, and yet we're getting it for free out of the air and we're doing nothing about it. So there are so many simple things we can do. And while you're on water, and I was talking to Padraig on this, like, Jesus, when you're traveling for the last week, you really have so much pent-up aggression. So apologies in advance. But you know, here's the scariest thing of it all. And there's a load of biopharma plants in Ireland that use Triton X, which is a restricted substance, right? It's a substance of very high concern. So you have to be really careful on how you dispose of it. They burn 99.5% pure water because it has residue of Triton X. Now, there's a challenge to all the great brains in PMTC. Come up with a cool piece of technology to take that out. They've looked at the likes of the charcoal filter that Rachel and Andy were on about. Doesn't work. There's your challenge, because not only is it a really cool engineering challenge, it's a tiny market, so the big majors aren't interested in it. It will be very, very distinct technology when you get it. You'll have a monopoly. It will be a small market you'll have a monopoly on, and you'll be very easy to scale it subsequent to that.
Good. What's your percent for that?
Well, it could be in for a little bit.
Thanks, Michael. Okay, well, there's an interesting, we can follow that one up when we get out. Good, let's move it on a little bit. I mean, the graph is interesting in terms of the, I suppose, the percent of yes, which is at 30% of companies. So I'm guessing, and no, I'm more than guessing, I'm quite sure that AstraZeneca is within that 30%. So maybe moving on to, I suppose, looking at the topic of designing the plans for the future and the considerations and the implications of taking sustainability into account in making decisions to invest, and then the actual technology. Maybe, Rachel, you have some thoughts based on your involvement in the new project and lessons from the old. I know you touched on it earlier, but maybe just to give it a bit of thought there.
Yeah, sure. So, as you're all probably aware, AstraZeneca have the net zero carbon ambition for 2030. So we are seeing that within the design of the new build. We have the facilities and fleet kind of targets, and we're designing the new facility to be up to LEED Gold certification. So part of that, we've seen solar farms on the roof of the production building. We've got green gas supplies to the site. We're looking at, sustainable things in terms of the facility, which, I'm not involved in that, so I can't talk too much to it. But in terms of like the process sustainability, because of the nature of the facility, it's a multi-product plant. We've looked at solvent recovery, we've looked at heat recovery, but for some heat recovery instances, you need a unit running 24/7 to really get the benefit out of it. So in that instance, the capital investment, it's not going through because it's hard to identify a unit where you can generate that heat recovery. And then in terms of solvent recovery, there's a wider issue of you can't use solvent from product A on product B, for instance. So if we take that solvent recovery offline, we've probably finished the campaign of that product before we've even recovered it. So we have thought about all these solvent recovery systems, heat recovery systems, from a sustainability perspective within the CapEx budgets for the projects. But when it comes down to the actual overall budget for the project, we've had to take those out. But we are leaving space for them in the future on the external of the facility for a solvent recovery system. So yeah, it's a tricky one in terms of CapEx, because we are finding when we're limited on budget, they're the first things to go for the sustainability initiatives from a process perspective. The facility side, they're all kind of key for the first part of the roadmap of our...
And is part of that this potential conflict with the GXP aspects and this risk of product cross-contamination as it used to be? So is there a technical problem to be addressed there in terms of how can you really bring a recovered solvent back to the point where it could be treated as virgin solvent effectively?
I think so, yeah. So with solvent recovery, if you can get it back to the raw material spec, technically it's raw material spec, so you could technically use it, but there will always be that risk of trace API from one product to the next. Yeah. We could see the potential argument of reusing the cleaning solvents on other product cleans first, but the actual process solvent recovery, I think we're a bit away off that yet. Yeah.
And then maybe Andy, bringing that on a little bit, what was the specific driver towards the cleaning project? I mean, it came out of one of Astra's goals to the net zero goal, but what was the challenge? I think you mentioned previously there was a competition, an internal competition, and you put it forward and it won on some grounds.
So we looked at the cleaning process purely from our own initiative, wanted to reduce the volume of solvent that we were using. It was then we were made aware of our department's PTD sustainability challenge and this pot of money from the NNRG fund. And that's a challenge that's extended to the whole of our department, and it calls for business cases for potential projects. You then present a business case, and the winning entries are then applied, have funding applied to those projects.
So within AstraZeneca, a dedicated group was set up to drive, if you like, the green agenda and the goals. Yeah. And do you think that that's something that is working, and is it something that companies should be thinking about, having a dedicated resource to drive these things forward and create challenges or drive innovation?
It's over. It receives an awful lot of applications every year. We pride ourselves on being entrepreneurial, and it drives science, people following the science. And it's almost seen as a competition within areas to drive sustainability and who can achieve the greatest outcome.
Very good.
Do you know what I thought was great about the example Andy and Rachel gave? Because it showed you, and I think when you're in academia, until such time as you work in a chemical plant, you don't realise how much of what goes on there is cleaning. Like if you don't have a clean plant, you don't have a clean product, so you can't sell it and you can't inject it or administer it to patients. And what I loved about that was when you came to your last slide and you showed the results of the API 1 and API 2, and even though you saved more solvent on API 2, because the cost-benefit analysis didn't rack up, you don't run with it. So it doesn't matter how much solvent or how much whatever it is you're saving. Pharmachem companies are in the business of making money, and they should never apologize for that, because all of our pension funds have invested in the pharmachem companies, and that's what's going to sustain you when you retire. But that's what they do. They don't do it. Don't be lulled to think they do it for the good of humanity. They'll do it because they're making money. And if they can do it for the good of humanity while they're making money, they will do it willingly. And that's said in a very constructive phrase. But that's why people who come up with fabulous ideas, if you're going to be burning through charcoal filters at a rate of knots, it will never get done. And that's what's lovely about that project. You saw some fabulous metrics. You saw some great innovation and some great clear sky thinking almost. And then it came down to, we're going to run with this because it makes money for us. I loved that.
Very good. Well, that leads on to the metrics discussion.
Question for the audience, maybe a polling question. Does your company monitor its site energy in real time or based on review of historical data? And I suppose this comes back to one of the points that John made around, what are you measuring? Because if you're not measuring, you can't drive performance. So we just wanted to get a flavour of that, just to see what's existing, if you like. It again helps us to understand, okay,
yeah,
so energy is in real time.
Well, let's see how it evolves over
Not
time.
sure.
I mean, I think, I suppose the reality is, when it comes to electricity, a lot of companies still would review their bills and they'll look back over the last 2-month period and they might plot them and look at it compared to last year and that. But and this touches on the topic of metrics and reporting, I suppose. But first of all, the metrics side of things. What are the things that we need to be measuring more real time than historically in order to get a true picture of what's happening today? What are the metrics that we need to be drilling down into to see what's happening at the equipment level as opposed to seeing at the site level, for example? And then how should these be analysed and reported? And I was thinking, John, there maybe you'd have some thoughts on where there are opportunities for companies as to what they can be measuring and how much more detail using real time.
Yeah, I think the approach generally in industry has been to leave energy to the facilities team, and so they're looking at it through one particular lens, which is just about reducing cost, and it's often quite sort of siloed. And particularly on very large sites, it's about optimizing maybe the maintenance over the actual energy usage. But if you can bring your metrics down to the level of the product family or the particular batch or the particular building or lab, then you can actually move those metrics to being the responsibility of the production operation. And you can have comparisons between different product families or between different buildings or between different product lines. And you can wrap up all of that, the water, the air, the gas, electricity, steam. We've seen examples in plants where, for hand washing, they were mixing steam with chilled water because both were available next to where they wanted to have a sanitary facility. So, an utter waste of the use of both. And and in another example, in a pharma plant actually that I won't mention, a batch process needed a specific controlled environment, but only for 3 days out of the 10 days. But of course, it's easier to control the environment for the 10 days and do that. And also that building had 3 floors that only needed the controlled environment in one section. But again, the building was built with the controlled environment in the entire building. So a very, very quick analysis of linking the actual energy consumption to the product being built, really showed up opportunities that would have been missed in a plant-wide audit. So we call them zeros, significant energy reduction opportunities, and they're the ones you want to hunt. They're the 80%, the least amount of effort for the greatest amount of saving. And it's identifying those and having a roadmap of reducing them. I think you can gain some significant wins.
Good. Do you guys have a lot of real time reporting in Macclesfield, or is it being built into the design in the new plant, or is it something you're looking at?
Yeah, because of the age of the assets in DSM, I don't think we do any monitoring, do we? But with the new facility, we are looking at kind of the smart building view. So where areas aren't being used, HVAC consumption could be reduced, and where labs aren't being used, lights will be turned off, all those kind of links. And I think it's the BMS link into the EMS and all that smart technology stuff that we're building into the facility. And then in terms of like process water reuse, you were mentioning earlier, rainwater harvesting toilets, that's one thing we've got. So yeah, we're trying to build all those sustainability things because we're starting with a blank page. It's kind of, we have to put that in from day one.
Yep. Very good. I suppose there's always the issue in pharma is we have a process that works, and if we have to change it, it's going to cost a fortune. The regulatory department won't let us. But what we can see coming is, based on what you're saying, Michael, there are going to be some real conflicts where some materials that we've been using on
are going to be gone.
Are going to be gone.
Yeah. They're going to be gone.
And I suppose what advice do you have for companies to be sure that they're clued in from a horizon point of view as to what's coming, what is coming, so that they can give a heads up to their site leads or whoever?
I think actually how Andy described it in terms of the team they have in AstraZeneca that funds innovative thinking in terms of sustainability. It can drive so much above and beyond what you do in a pharma company, because with the best will in the world, they tend to be such large conglomerates at this stage, and there's so much silo thinking. I'm in tech transfer, that's what I do tech transfer. And you don't necessarily extrapolate it on to when it goes into the plant. I've come from a 30-year-old piece of kit, I'm going into a brand new piece of kit. And you're not necessarily thinking how this can be improved upon. You have the process, that's how we've done it, that's what we filed. This is what we're going to do. There's greater flexibility required in the next decade in terms of how we transfer technology from one plant to another. Because, you know, even if you look at it in terms of batch size, and I think it was Emmett's presentation was talking about the greater potency of drugs, and or maybe it was David's, the potency of drugs in the smaller quantities. Batch sizes are shrinking all the time. Very few are single product plants anymore, so the need for adaptability is huge. So the thought process internally in companies is going to be like it never was before. And it's interesting, if you look in Ireland, the number of plants in the last 10 years that used to be a dedicated plant and through takeover or whatever have become CDMOs. That's where you're actually seeing real innovation coming from the CDMO community, because they have to compete to survive. And for those who aren't familiar, these are the contract development and manufacturing organizations who often make things on contract for the big multinationals. But they have to be so responsive. They're actually driving an awful lot of innovation. And like, I think, Rachel, you said you're looking at solar panels on the roof in the new site in Blanchardstown. I think that'll become common in the next 5 years. And in fact, during the week I saw where the French government is bringing in a law that all car parks with more than 85 spaces have to now have solar panels on it. And it will be the equivalent of 10 nuclear power plants. So like, think of the innovation that don't go for incremental change, go for big change. Go for big change and you'll actually drive it. And I think the companies who do that best will thrive the most.
Very, very good. I think
we're coming up on time, but I think you're right. And you know, you've mentioned it, Padraig, that AstraZeneca have made commitments around net zero targets. They're not the only ones. There's quite a number of pharma companies who've done that. And so, you know, hopefully this is an area where pharma is leading the way. And, you know, there's plenty of low-hanging fruit, whether that's in the energy side, in the process side. And I suppose the job for us in PMTC is to drill a little bit deeper. So I mentioned at the start of our afternoon session, we plan to have a workshop in March, and I know a sheet has gone around. I've seen a few people have signed up. So for anyone online or here in the room, if you have an interest in that, we'll assemble another group and maybe a wider group, and we'll get into a much more dynamic workshop type discussion, because I suppose, you know, very dynamic discussion and really engaging. And I suppose, look, for all of us as citizens, whether we're in this industry or not, I think this is something that everyone has to grapple with. So hopefully we can be part of that solution, and we look forward to future discussions on that. So with that, I'd like to say thank you to all of our panel members. I think it was great kick-off to this debate, which I think we're going to continue, and open invitation to everybody to come to that session that we're going to have in March. So thank you very much. I should have asked if anybody has any questions. Maybe I was a bit premature there. So before folks step up, is there anybody, I don't see any questions online, but anybody in the room that wants to make a point or ask a question before I announce the winners of the poster award?
Do you envisage any kind of alignment with the drug regulatory agencies, with the kind of sustainability projects that might come in terms of how drugs are manufactured? Is there any kind of meeting point in terms of companies that are willing to make changes to how they manufacture their products and how that aligns with drug regulations? Yeah.
Zero alignment. And in fact, we're meeting with the HPRA, as Padraig knows. Within BPCI, most of my engagements are with the HSA and the EPA. And I've mentioned this REACH reform, and one of the things in the REACH reform is a concept of essential use. And basically, if you're using substances of very high concern, it's only when the medicine is an essential use medicine. It's not been defined. So it says essential use for healthcare. So your acne treatment, is that essential use? Maybe it is, maybe it isn't. Your statin, is it essential use? Because all it does, it lowers cholesterol. Does cholesterol actually kill you? No, it's one of the 5 parameters they look at. So all of a sudden we go into the debate in terms of essential use. And this has been driven by DG Environment and to a lesser extent DG Grow in Europe. And DG Sante have not been consulted yet. So I know this has been talked about for the last 18 months, and we're going in to talk to the HPRA the next day and say, listen, any chance you can use your contacts in the European Medicines Agency to say, listen, can we actually have a look at what you're determining is an essential medicine? Because, hey, we regulate medicines. I think we're the best people placed to talk about it. But currently, there is zero overlap.
Sobering thought to end on.
Sorry, I mean, there is a point there. In your application for a new medicine authorization, you are obliged to perform certain environmental testing with regard to what happens to medicine after the patient, basically when it's excreted, where it goes, how it's treated in biological plants, where it ends up in the environment, that sort of thing. That assessment is done, but it's not clear what they do with that information and what decisions are made on the back of it, because I'm not sure a drug has been turned down because of it, maybe a poor result on that one. So, but I guess the point is that in law, the impact on the environment is something that's taken into account. Yeah. So somebody could come in and tighten up on that and say, well, you've been measuring it for years and haven't been doing anything, so we're going to tighten up on it, and then you'd see the impact, which might affect, for example, how a drug is delivered as opposed to whether it needs to be taken at all. So.
And that's the irony. It's considered from a health downwards, but it's not considered from a chemicals upwards. So when you make the new drug application, you do have to put in environmental consideration. But when DG Environment changes REACH, they don't consider the medicine. And that's the scary piece, because we use less than 1% of the chemicals in Europe.
So clearly a lot of dialogue needed. So I think we're in good hands, Michael. If you're the man who's going to go and champion for this industry, I'd have to say, and you're a wealth of information. So it's somewhat of a sobering thought, but we'll turn it into a positive. Loads of opportunities there, right? Loads of opportunities there.
Loads.
So look, I think it's a huge topic, as was the data analytics. That gives me comfort, because I think we're going to be a busy centre for the foreseeable with the help of God. And so I mentioned again earlier that we have a poster display, and hopefully many of you got to go and have a look, and likewise with the exhibits. And first to say, we voluntold a number of our stakeholders to act as judges today. So I'm going to call out to Dr. Sandra Lennon from MTU, who was one of our poster judges, Dr. Marion McAfee, ATU, who was a poster judge, Dr. Liam Lewis from Kappa also acted as a poster judge, and Kenneth Pearce from Hyde, the same. So I'm delighted to announce that we have winners of that competition. In third place, I'd like to welcome Victor Simon to come and collect his award. Okay, I know folks are probably anxious to get on the road, so without further ado, in second place, we have Stephanie Cassilligan. So Stephanie, if you'd like to come down and accept your award. Okay, and last but not least, again, one of our own, and this wasn't a fix, so I did name the judges in advance. I'd like to welcome Dr. Nicholas Aramouni, first prize. So I think from our point of view, really, really successful day. Thanks once again to everybody who came today, both in person and online. I think we still have some people online. You know, I hope you'll agree it was a great knowledge sharing day, which is really what our objective is. But it was also great to get some cues and insights in terms of next steps. So we'll be in touch for that workshop that I've talked about. Thanks once again. Safe home to everybody, and do keep in touch. Thanks so much.